Okay, so first of all, I'd like to thank Niels and Lauren, the organizers of Microsymposium A93 on nucleation and crystal growth. I'd like to thank them for their kind invitation to give this lecture, and also to thank them for the opportunity to present a recording of the lecture when it became clear that I would be unable to travel to Melbourne at this time. And I'm very sorry to be missing the IUCR Congress in person on this occasion. What I'd like to discuss during my lecture is new in-situ NMR techniques that we've been developing to study the progress, time-resolved monitoring of crystallization processes. I want to give some background to these techniques, various examples of polymorphic evolution, new developments of techniques, and further applications through the lecture. Okay, so the basic idea is very, very straightforward. We want to monitor the formation and evolution of the solid phase as a function of time during crystallization from solution, typically. And the main idea, or the main aim, is to understand the sequence of crystalline polymorphs produced on the pathway towards the final crystallization product. Clearly, a basis for being able to use solid-state NMR in this context is the fact that solid-state NMR techniques can readily distinguish different polymorphs of solid materials. Here's just one example of a polymorphic system, sodium acetate, and we're showing on the right-hand side solid-state carbon NMR spectra, 3 known polymorphs, and we can very clearly distinguish these 3 polymorphs from the isotropic chemical shifts observed in the solid-state NMR spectrum. So solid-state NMR is a very powerful technique for identification and characterization of polymorphic systems, as shown here. Okay, well, let's discuss in a bit more detail the in-situ solid-state NMR strategy. And that idea is very, very straightforward. We basically set up a crystallization experiment inside the solid-state NMR spectrometer. Usually we induce crystallization by a rapid decrease of temperature, and then we simply record solid-state NMR spectra repeatedly as a function of time during the crystallization process to monitor how the solid phase changes as a function of time. Here's a cartoon of what we're doing in this type of experiment. So let's start with an undersaturated solution at high temperature. We cool rapidly, it becomes supersaturated, and we might then see the beginning of crystallization. And all the time we're recording the solid-state NMR spectrum, and here we can see the emergence of the solid-state NMR spectrum, characteristic of a particular polymorph shown in red. We may see then the growth of that polymorph corresponding to an increase of intensity in the solid-state NMR spectrum, but then one can observe polymorphic transformation to a more stable polymorph shown in green, and now the more stable polymorph grows at the expense of the original one shown in red. We can see all these changes occurring in the solid-state NMR spectrum, and therefore we can deduce the changes in the identity and the amount of the different solid phases present as a function of time. So one question that we should answer is, what's the advantage of using solid-state NMR for in-situ studies of crystallization? As we know, many scattering techniques, different diffraction techniques, a wide variety of spectroscopic methods can be used to study crystallization in situ. But I want to emphasize there's one big advantage of using solid-state NMR, and that was really the basis for us focusing on solid-state NMR in this context. And the big advantage is that we can set up the measurement conditions such that we detect only the solid phase, and the liquid phase, the solute dissolved in the liquid phase, and so on, are actually invisible to the measurement. So we can set up the NMR measurement so that we selectively detect only the solid phase. And that can be very advantageous in allowing us to study early stages of crystallization where the amount of the solid phase is very small and most of the solute, most of the molecule of interest is actually present in the liquid phase. But we focus in only on the solid component of the system of interest. And let's just illustrate that using this example to illustrate the selectivity of the measurement, how we can detect only the solid phase. So here's a heterogeneous liquid solid system. It's a saturated solution of glycine in water. And this is the NMR spectrum recorded under normal direct polarization conditions. And we actually detect these very sharp lines for the two environments in carbon environments in glycine, but also we see a very broad line here, and that's the solid phase of the carboxylate carbon environment. And just zooming into that in more detail, we can see the part that we want to observe, the signal from the solid phase, but superimposed on that, it's a huge signal from the dissolved glycine in the solution phase. However, by recording the solid state carbon NMR spectrum, or using a different pulse sequence to record the NMR spectrum, so-called proton to carbon cross polarization, this pulse sequence is selective only to measuring the solid phase. So for the same sample here, we can see only the signal from the solid, and we've completely eliminated any signal from the dissolved glycine in solution. So this gives us selectivity in measuring only the spectrum from the solid component present in our crystallization system. So now, in terms of time resolution, which is a very important consideration in any in situ experiment of this type, the time resolution of the study depends on the time to record a single solid state NMR spectrum of adequate quality to allow us to determine what polymorphic forms are present at a given point in time during the crystallization process. And clearly, the quicker we can measure a suitable solid state NMR spectrum, the better the time resolution in monitoring the crystallization process. So we want to be able to record the spectra as quickly as possible. We want, therefore, to maximize the sensitivity of the measurement, and that might, in the case of carbon-13, involve isotopic labeling, but we always benefit in this regard by carrying out measurements at high magnetic field. And the vast majority of our work in this field has been carried out at the UK National High Field Solid State NMR facility, which is based at the University of Warwick. 20 Tesla magnet, 850 megahertz proton, resonance frequency, and here's this very large NMR magnet and the spectrometer where we carry out these measurements. Okay, well, let's move on to some examples of studying polymorphic evolution using in situ NMR. And let's focus, first of all, on crystallization of glycine, which is a very well studied polymorphic system. There are three known polymorphs at ambient pressure called alpha, beta, and gamma. Here are the three different crystal structures, and the relative stability, gamma is the thermodynamically stable polymorph, alpha is a bit less stable, and beta is significantly less, the least stable polymorph among these three. But fortunately for us, we can clearly see a difference in the isotropic carbon chemical shift for the carboxylate carbon environment in these three polymorphs, as indicated here. So that allows us to very readily determine which polymorph is present at different stages of time during crystallization. So the first experiment we carried out was crystallization of glycine from water. And this, here we see the carboxylate region of the carbon NMR spectrum as a function of time between 0 and 14 hours in this experiment. And in the duration of this experiment, we simply see the formation and the growth of the alpha polymorph. There's actually no polymorphic evolution, and this agrees with literature on crystallization of glycine from water. However, we also tried the crystallization of glycine from deuterated water. And look at what happens in this case. We see initially the same as in natural abundance water, the initial formation and growth of the alpha polymorph, but then after about 2 hours, we see a decay of that signal and the growth of a signal characteristic of the gamma polymorph. So we're seeing polymorphic evolution in this crystallization system. And we can quantify the relative amounts of the alpha and gamma polymorphs. This is not necessarily straightforward. We need to calibrate the relative cross-polarization efficiencies of the different polymorphs. But once we've done that calibration, we can get data like this, showing the relative amounts, the growth of the alpha polymorph, then after 2 hours that decays, and that is mirrored by the growth of the gamma polymorph, as shown there. Crystallization of glycine now from methanol water mixture, and here we see a very different crystallization pathway. Here we see in the very early stage of crystallization, the growth of the beta polymorph, but that decays quite rapidly and is replaced by the alpha polymorph, a transformation from beta to alpha. This shows us, you see, if we want to isolate this very unstable beta polymorph of glycine, what we need to do is start the crystallization and stop it immediately, because at this point in time, we have essentially a pure sample of the beta polymorph. And this is one of the key aspects of this technique. It gives us information on the time window during which specific transient polymorphs, metastable polymorphs, are present during crystallization experiments. And here's a summary of the situation for glycine in the different solvent systems and the different polymorphic evolution deduced from our in-situ NMR technique. So glycine is very commonly studied organic polymorphic system, and an extremely commonly studied inorganic polymorphic system is calcium carbonate. So I want to discuss some of our results on calcium carbonate. 3 crystalline polymorphs: calcite, the thermodynamically stable form; aragonite; and vaterite, the least stable form. And there's also an amorphous phase, which is known and also believed in many cases to be a precursor to crystallization. And again, there are clear differences in the carbon-13 chemical shifts for these 3 different crystalline polymorphs, allowing us to readily identify them and detect them in the in-situ solid-state NMR experiments. But here's also the very broad line characteristic of the amorphous phase of calcium carbonate. We've carried out these experiments by mixing aqueous solutions of calcium chloride and sodium carbonate with carbon-13 labeled carbonate in all these experiments. And we've studied the crystallization under different environments. I want to describe in-situ NMR of crystallization inside agarose gel. Okay, so here's the results for that experiment. And what we see is actually a mixture of vaterite and calcite. So at the very beginning, we have a predominance of vaterite, but also some calcite present. And as the crystallization progresses, we see polymorphic evolution. The vaterite transforms to calcite. And by the end of this experiment, we have essentially a pure phase of calcite. So again, monitoring the polymorphic evolution. Now, in the presence of some amount of magnesium, this is known in literature to favor the formation of aragonite, and indeed that is what we see in this experiment. But at the beginning, we see this very, very broad line in the solid-state NMR spectrum, characteristic of the amorphous phase of calcium carbonate, and that then transforms, that then crystallizes to form the aragonite polymorph as shown there. So again, very clear information on the evolution of the different polymorphic forms during crystallization, and also with the intervention of an amorphous phase in that particular case. So in the case of glycine and calcium carbonate, all these polymorphs were already known prior to our experiments, but we also find cases where we discover new unknown polymorphs transiently during crystallization. And one very quick example here is the co-crystals formed between urea and dihydroxydecane in this case. So for this particular system, there is one known co-crystal, the structure of which is shown there, hydrogen bonded arrays, as one would expect. But when we carry out the in-situ NMR study of this system, and this is done for carbon-13 labeled urea, so we're focusing here on the carbon NMR signal for the urea environment. What we can see here is initially we have a single peak for urea at this position, and then after about 40 minutes or so in this experiment, we see a very sudden change to a peak in this position, which is characteristic of the known co-crystal. And just to look at the 2 individual spectra, so in black is the final known co-crystal, but what we can see is a transient polymorph or a transient solid form here with a very different chemical shift for urea. And this leads us to the discovery of this new phase in this particular system. And obviously, because we can see from the in-situ NMR exactly the time period during which that transient polymer or that transient solid phase is present, then we can stop the crystallization, isolate it, and characterize it from this knowledge, from these results. Okay, well, what I've described there were our early studies where we focused on measuring the solid state NMR spectrum and mapping the changes as a function of time during crystallization. What we then realized is there's actually a much more informative way of doing this experiment, and this is called classic NMR. So classic doesn't mean that it's old or ancient. Classic means combined liquid and solid state in situ crystallization NMR. Because what we recognize is that during the crystallization process, as the solid phase is produced, there's actually lots and lots of changes occurring in the liquid phase. And what we really want to be able to do is to monitor both the evolution of the solid phase and the evolution of the liquid phase at the same time. And this is what classic NMR allows us to do. And what we do here is we have two, or in some cases more than two, but at least two different pulse sequences. One allowing us to measure the liquid state NMR spectrum selectively, and one pulse sequence allowing us to measure the solid state NMR spectrum selectively. And we simply alternate between these two different pulse sequences during the crystallization process. So this gives us insights into time-dependent changes, both in the solution state and in the solid state during the same crystallization process. The way we set up the experiment is as follows: two different pulse sequences. The one in red, phosphorylation, for example, selective to only to detect the solid phase, and another pulse sequence, shown in blue, selective to detect the liquid NMR spectrum. And during the crystallization experiment, we simply alternate the measurement of the solid state and liquid state NMR spectra, alternate between the red and the blue pulse sequences, as shown there. And then at the end of the experiment, we separate these into a time result set of solid state NMR spectra and a time result set of liquid state NMR spectra, telling us selectively about the evolution of the solid phase and the evolution of the liquid phase during the same crystallization experiment. So the sort of information we can obtain from the solid state is similar to what I discussed before. Changes in the amount of the solid phase, changes in the polymorphic identity as a function of time. And for the liquid state NMR, we get information on things like the change in concentration as a function of time, changes in solution state interactions, changes in solution state speciation, and so on. Because we remember that, as the solid phase is produced, the liquid phase gets more and more dilute, and therefore all the interactions will be changing. And this is what we want to capture, the complete understanding of what changes in both the solid phase and the liquid phase during the crystallization process. So let me illustrate the sort of information we can obtain by a study of crystallization of meta-aminobenzoic acid from DMSO. This is a carbon NMR study in both the liquid and the solid phases. So meta-aminobenzoic acid has 5 known polymorphs shown in the carbon NMR spectra of the 5 solid phases are shown here. And first of all, we note that 3 of the polymorphs contain the zwitterionic form of the molecule, and 2 of them contain non-zwitterion, as shown here. We can very clearly identify from the solid state carbon NMR spectra which ones are zwitterionic, so they contain a peak here corresponding to this green dot, whereas the non-zwitterionic polymorphs contain a peak in this region shown by the red dot. Okay, now let's look at the results from our classic NMR experiment. And here's this evolution of the solid phase, the evolution of the solid state NMR spectrum as a function of time during the experiment. First of all, we notice that for the first 2 hours of this experiment, there was actually no signal in the solid state NMR spectrum. And that is telling us that what we have at this stage is a supersaturated solution that, is thermodynamically unstable It wants to crystallize, but it is just waiting for that favorable nucleation event to happen before the solid phase is produced. So we have this induction time before crystallization happens, but once crystallization does happen, we see the formation of form 1, and actually there's no polymorphic evolution in this case. Form 1 exists and simply grows in amount as a function of time during this process. We can look at the integration of the solid-state NMR spectrum and see no signal at the beginning. Then after 2 hours, we see the growth of the signal in the solid-state NMR spectrum, indicating an increase in the amount of the solid phase. But correspondingly, we see after 2 hours a diminution in the signal intensity in liquid phase, and that corresponds to the loss of the MABA from solution as it is as the crystallization occurs. Okay, but we can actually get much more information about changes in liquid phase by looking at how the chemical shifts for all the carbon environments in the molecule and in the solvent, to look at how these chemical shifts change as a function of time during crystallization. So we're going to zoom in on each of these carbon chemical shifts. We'll look at them over the same chemical shift range and see how they evolve as a function of time. So focusing first on carbon 6, we can see, look, at the beginning, for the first 2 hours, no change in the liquid phase chemical shift. But then suddenly, at the moment when crystallization begins, at just about 2 hours, then we can see a very well-defined change in this chemical shift to a lower value, as shown there. Looking now at carbon 3, this one here. Again, no change for the first 2 hours, and then suddenly, at the moment when crystallization begins, we see an evolution of this chemical shift to higher value during the crystallization process. And indeed, each of the carbon environments shows no change in the first 2 hours and then evolves in a very well-defined way, and indeed at the same rate for each of these environments, as shown there. And even the solvent shows a very significant evolution in chemical shift as a function of time once the crystallization has started. And all of these changes in liquid state chemical shifts tell us there must be changes in the interactions and the environment of the molecule in solution, and maybe also the speciation as a function of time once the crystallization is actually progressing. Okay, well, how to interpret all this information? Let's put it all onto one graph as shown here. This is the change in the carbon chemical shift relative to the value at time 0. So the carbon NMR, all the chemical shifts stay essentially constant for the first 2 hours, and then they all change in very well-defined ways as a function of time during crystallization as more and more of the solid phase is being produced. So to interpret this, what we remember is that in the first 2 hours, before the crystallizations happen, we have a pre-nucleation supersaturated solution, thermodynamically very unstable, is wanting to crystallize, waiting for nucleation to happen. But what we have then, once crystallization happens, the solution phase decreases in concentration and eventually reaches an equilibrium saturated solution, which is thermodynamically stable and does not change anymore with time. So to interpret all these changes, what we're going to do is characterize the final solution. This can be studied independently by NMR and a wide variety of other techniques. It's a stable solution. And then what we're going to do is interpret all of these changes working back in time to give us insight into what this pre-nucleation supersaturated solution is like. Very poorly understood stage of crystallization, thermodynamically unstable solution, but we can interpret all these changes going back in time to give us insight into the nature of that solution. Okay, so to summarize our conclusions then, on doing that, what we have is as follows. So after crystallization, we know that we produce form 1, and that's a zwitterionic form of meta-amino benzoic acid. We also know from the equilibrium saturated solution that this comprises non-zwitterions for MABA and DMSO. And what we can tell from the changes in the liquid state NMR spectrum going from here and working back in time, this tells us that compared to the equilibrium saturated solution, the pre-nucleation solution contains an increased proportion of zwitterions and or aggregates containing non-zwitterions engaged in OH-N hydrogen bonding. And these are very, very plausible precursors, for example, pre-nucleation clusters for the ultimate formation of a solid phase containing zwitterions. And that's the sort of information we can deduce from this technique. Okay, well, let me give you two more examples of classic NMR. One is crystallization from the liquid phase of DL menthol. So here we melt the DL menthol, we cool gradually and monitor the solid state NMR spectra, the solid state NMR spectrum and the liquid state NMR spectrum in the classic NMR experiment, alternating between the measurement of the solid state NMR and the liquid state NMR spectrum. So here's the results. On the left-hand side, liquid state NMR as a function of time during crystallization, and on the right, the solid state NMR spectrum as a function of time. So what we can see is for the first 30 minutes or so, we only have a signal in the liquid state NMR, no signal in the solid state NMR. And then very suddenly, at this point of time, it's as if the whole liquid phase solidifies, and then we can see what we have here. So we see a signal in the solid state NMR spectrum, but let's zoom in more detail. And the initially formed solid phase has very broad lines, as we can see here. And that's characteristic of amorphous phase. So that's the first solid phase produced. And then at about 50 minutes, we then see the production of a crystalline phase. The broad line amorphous phase then suddenly changes to much sharper lines, characteristic of a crystalline phase. And here, just to look in more detail at some individual spectra. Here, the liquid phase, here, the amorphous solid phase, and here, the ultimate crystalline phase. And we can see the very broad lines characteristic of an amorphous phase, and the final, much sharper lines in the crystalline phase. And comparing to the known spectrum for the alpha polymorph of DL menthol, we can identify the final product as the alpha polymorph from this experiment. I want to also discuss some recent work on monitoring the formation of a metal organic framework by In-Situ NMR. So again, this utilized the classic NMR experiment to study the formation of this MFM 500 MOF containing nickel and containing this linker shown here. So we carried out a classic NMR experiment, but what I want to focus on is our in-situ liquid state proton NMR spectra as a means of getting very detailed information on the kinetics of this MOF formation process. So here's the proton NMR spectra as a function of time at different temperatures from 60 degrees centigrade to 100 degrees centigrade. So there's a lot of things happening in these spectra, but I want to focus on these three peaks shown here. So these are the peaks in this part of the spectrum at 60 degrees and how they change with time, because these represent the three different proton environments in the linker. And as the MOF is formed, these peaks will decrease in intensity as the linker is consumed from the solution and is utilized in the formation of the MOF. So we're going to look at the decay of these three peaks as a function of time in each of these experiments at different temperatures. And what we can see here for the three different proton environments in the linker is a very well-defined decay as a function of time. And in each case, these have been fitted using the Gualtieri model for nucleation growth, which is a commonly applied kinetic model for understanding solid state formation processes, crystallization processes. And in each case, the experimental data for all three proton environments has been fitted by the Gualtieri model, but using a single set of parameters at each temperature. And I hope you agree that these represent fairly acceptable quality of fit. So what are the parameters that we fit in this case? So from the Gualtieri model, we get the rate constant for nucleation, the rate constant for growth, and the standard deviation in the mean nucleation time. And looking at some of these parameters, so from the standard deviation at mean nucleation time, we can get information on the probability distribution for nucleation. So we can see at high temperature, very sharp probability distribution at an early value of time. At low temperature, much broader probability for nucleation, and the percentage at a much longer value of time, around 11 hours in that case. And then from the rate constant for growth and rate constant for nucleation, we can deduce activation parameters from Arrhenius plots of this type. And I hope this has shown you we can really get quite detailed kinetic information on crystallization processes, both growth and nucleation, from the in-situ NMR data. Finally, I want to briefly discuss crystallization from a frozen solution. And here we're going to focus on the formation of glycine dihydrate from a frozen solution. So I mentioned earlier the three anhydrous polymorphs of glycine, and it was only in 2017 that a hydrate phase of glycine was actually discovered. And this is glycine dihydrate, discovered by Tony Hu from New York University and his co-workers. And what they did was a powdered diffraction study where they quenched a solution of glycine in water to low temperature and then gradually warmed to about 210 Kelvin and found this dihydrate phase of glycine, the structure of which they determined from powdered diffraction data. So we, with our collaborators from Marseille, we studied the same process in situ by solid-state carbon NMR. So we quenched a solution of glycine in the water, and I should say here we're using carbon-labeled glycine labeled in the carboxylate carbon environment, quenched at low temperature, and here we can see the amorphous frozen solution at 165 Kelvin, but then gradually heated it, and then at about 211 Kelvin in our experiment, very close to the temperature that the previous study, hydrorectory diffraction study, observed the formation of glycine dihydrate. We see the appearance of a sharp line characteristic of a crystalline phase. Now this crystalline phase, has this particular isotropic chemical shift, and that is different from any of the three anhydrous crystalline phases, alpha, beta, and gamma. And the glycine dihydrate appears here. And because this solid phase is produced here, it doesn't evolve at this temperature, we can actually carry out fairly sophisticated NMR experiments to characterize what material we have. Head core here is completely consistent with it containing water, for example. And this measurement, the carbon dipole buildup, so the diamonds here are experimental data points in this type of measurement for the material produced here. And the dashed line is what the calculated dipolar buildup would be like for the known crystal structure of glycine dihydrate. So this very, very good match between experimental and calculated data gives strong verification that the material that we've obtained in our in-situ NMR experiment is actually glycine dihydrate corresponding to the published crystal structure. So that really brings me to the end of my talk. I'd like to just mention that we're also collaborating with Julia Mollica and Pierre Toureau from the University of Marseille in France on the development of an alternative technique to study crystallization by dynamic nuclear polarization NMR. So this is solid-state NMR measured at low temperature, and these papers describe an ex-situ method for monitoring crystallization where we benefit from this significant intensity enhancement of dynamic nuclear polarization. Now, Julia is giving a lecture in Microsymposium A69 on Sunday, 27th of August, and I expect we'll be discussing this method during her lecture. So what I'd like to do is, I hope I've convinced you that in-situ NMR studies have a lot to offer in terms of studying crystallization processes, both to monitor the evolution of the solid phase, but also via the classic NMR technique to allow us simultaneously to study the evolution of the liquid phase. We've seen crystallization from solution, from the liquid phase, from frozen solutions, and also the use of these techniques to study kinetics of crystallization. I'd like to thank again the organisers very much for the opportunity to give this lecture, and also to thank all the people who've contributed to the experiments that I've described, members of my research group, research collaborators, and funding organisations. And finally, I'd like to request, if you've got any questions having listened to this lecture, please do not hesitate to email me, and I'll be more than happy to answer your questions. Thank you very much again.